Okay, so let's look at some past paper questions related to these areas. So a lot of times you're asked to define some of these important terms. So as we saw, allele are just the alternative forms of the gene. Then we had gene, which was basically the genetic code for a particular characteristic. Chromosome was basically a strand of DNA wrapped around histone proteins. And then we had deoxyribonucleic acid, which is the chemical that holds genetic information or instruction. Alright, then this says Jamal is a professional dog breeder. He mates, let me bring this up. He mates selected dogs with the intention of producing dogs with specific characteristics. He recently mated 2 dogs to produce dogs with desirable coat types. Coat type, so this is a gene, right? The gene for coat type in dogs is determined by a single gene with 2 different alleles. The straight coat allele is dominant and the silky coat allele is recessive. I'm going to make another video where I look at the crosses alone by themselves. So you could skip to that one if you need to see it. So name the process which leads to the production of dogs with desirable coats. So we spoke a lot about natural selection and how the environment basically allows for certain organisms to be better adapted than others. This one is artificial selection. We are human beings are the ones which are selecting who survives or which traits are passed on to offspring by selecting the ones that we find desirable as opposed to the ones that are most adaptable to the environment. So sometimes you might find that you select for a particular breed of dog, but that dog doesn't last long, it doesn't live long. It has very small bones that makes their bones more fragile, etc. They're not actually meant to live in that way. So that's something to keep in mind. Alright, so let's look at another question related to what we just looked at. So figure 2 is a series of randomly placed diagrams labeled A to E, showing the stages of mitosis in a plant cell. So as I recommend, whenever you have a diagram, before you jump in and start answering, assess the diagram. See what it is that it's actually showing you. So we know these are chromosomes. They look as if they're being pulled apart here. Here they look as if they are strands with this centromere here. Here they look like they are single strands. Here seems to be like they're starting to divide into 2 separate daughter cells pulling apart here. And here we see that they're lining up along the equator. So we already have an idea of what's happening, what comes first, etc. So we know how many, first of all, it asks how many chromosomes are in the parent cell of this plant. Now we can count either this one, whichever one looks like it's easier for us to count. Let's see, 1, 2, 3. So that one is not easy to count. So let's count this one over here. So 1, 2, 3, 4. And we can confirm that by looking at this one, pulling those sister chromatids apart. And it's 1, 2, 3, 4. So we could safely write 4 in the parent cell. Then, using the letter labels A to E, show the order. So first of all, we're going to have the single strands. Remember we spoke about them being single before they duplicate at B. And then they line up along the equator at E. And then they're going to be pulled apart by the spindle fibers, which are attached to the centromeres, which is the middle of the sister chromatids, at A. And then D is when they finally separated into their 2 daughter cells. State 2 reasons why mitosis is important to living organisms. Well, mitosis helps us to produce identical cells. That's the biggest thing. Identical cells which are needed for growth of tissues when you want to make the same types of tissues, or repair of tissues. For example, when you get a cut, etc., you're going to want to have the same type of tissue going back. Good. Now let's look at this one. Again, definition of gene, which we looked at already. It codes for a specific trait. Let me make sure that we did look at that up above. Yes. A genetic code for a specific trait or characteristic, allele, different forms of the gene or alternative forms, chromosome, the strand, etc. Now let's look over here. Figure 4 shows the process now of meiosis. So the one above here was mitosis. How do we know? Well, it says so. But also, we started out with 4 chromosomes, and as we count here, we ended up with 4 chromosomes in each daughter cell. So all of them ended up with the diploid number or the 2n number. Now meiosis is different in that in the figure 2 homologous pairs of chromosomes are shown in the original cell. So remember we said homologous means same, so you see these two are the same color and size, these two are the same color and size. One from father, one from mother, one from father, one from mother. Now observe the diagram before you jump into the question. What you see happening here at A? You see that crossing over that I was talking about, the mixing of genes. So the homologous chromosomes actually swapped some genes between themselves. Then at B, you see that the first division has taken place. How many chromosomes do we now have here? 2, as opposed to 4 in the beginning. Remember, even though they look joined up like this, this is still considered one chromosome because it's just an identical copy of the original one. So we see that we are already starting to have the haploid number or N number. And all of the all of the daughter cells so far are unidentical as opposed to in mitosis. And then by C, what we see happening here, again, the sister chromatids have now pulled apart from each other when they line up along the equator. And again, we still end up with 2 chromosomes in each, which is the haploid or half the number of the original 4. So with reference to figure 4, describe the events occurring at the areas marked A, B, and C. So what is the event happening at A? Well, at A, we see that we have crossing over occurring between homologous chromosomes. We also see that at A, there was duplication of the chromosomes as well. Duplication of chromosomes also occurred. Then at B, what is happening here? We have the formation of 2 haploid daughter cells. they are unidentical to unidentical identical to the parent. And remember, haploid means half the number of chromosomes as parent. Then what's happening at C? Now we have 4 haploid cells created at the end. So that's something also to remember, is that at the end of meiosis, 4 haploid daughter cells are the result, whereas in mitosis you have 2 diploid daughter cells, and these daughter cells in meiosis are unidentical. Because remember, we said that these are what are found in the sperm and the eggs, which are all unidentical from each other. That is why when you have fertilization happening, you never get 2 siblings, no matter how much the siblings are related, they will not look like each other exactly, because of that variation in terms of the sperm and the egg and that crossing over, mixing of genes. And last one we can look at here says figure 3 is an incomplete diagram of meiosis. 4 genetically, remember, 4 different daughter cells, P, Q, R, and S are the result. And again, we're going to observe how many chromosomes? 4. You see homologous pairs. These 2 are the same. These 2 are the same. Now, how they have them resulting? So we have 2 here, 2 here. Now, remember they would have duplicated first, right? So you would have had a duplicate come after this, a duplicate come after this, a duplicate here, and a duplicate here. So far, we only have one side of each. So all we're going to do is add the other side. Oops. Well, you realize I didn't really show the crossing over, so we don't need to show that either. So state whether the daughter cells produced by meiosis are haploid or diploid. So the daughter cells are always haploid for meiosis. Name one part of the body in which meiosis occurs. Well, definitely in the gonads, which are the testes and the ovaries. And then state the structure within the nucleus which contains the genetic material. The structure that contains the genetic material are the chromosomes. It is made up of the DNA. So that's that one. Looking at meiosis, mitosis, genes, and alleles.